Epigenetics. Epigenetics is a way of influencing how our genome is regulated without the DNA code itself being changed. Epigenetic can determine when genes are turned on and off, also referred to as gene expression, and which protein are produced as a result. It can even control the structure of the genome, relaxing the tightening package chromosome to allow the factor which control the gene expression access to the gene within. Without epigenetic, you wouldn't have developed from an fertilized egg to the multicellular organism you are today. And epigenetics will continue to impact on you, regulating specific gene in specific cell in the specific place at a specific time during your growth and development. Epigenetics is a study of how cells control a gene activity without changing the DNA sequence. Here, the epi means on or above in, and epigenetics describe the factor beyond the genetic code. Epigenetics change are modifications to DNA that regulate whether genes are turned on or off, and these modifications are attached to the DNA and do not change the sequence of DNA building blocks. Within the complete set of DNA in the cell, that is the genome, all of the modification that regulate the activity expression of the gene is known as epigenome. So because epigenetics changes help in determining whether the gene are turned on or off, they influence the production of protein in the cell. These regulation help ensure that each cell produce only protein that are necessary for its function. For example, protein that promote bone growth are not produced in muscle cell. Pattern of epigenetic modification vary among individual and different tissues within an individual or even in different cell within a tissue. Environmental influences such as person's diet, exposure to pollutants can impact the epigenome. Epigenetic modification can be maintained from cell to cell as cell divide and in some cases can be inherited through the generation. A common type of epigenetic modification is called DNA methylation. DNA methylation involves the attachment of small chemical group called methyl group. Each consists of one carbon atom and 3 hydrogen atom to DNA building blocks. When methyl group are present on the gene, that gene is turned off or silenced and no protein is produced from the gene. Another common epigenetic change in histone modification. Histone are the structural protein in the cell nucleus. DNA wraps around the histone, giving chromosome on their shape. Histone can be modified by adding or removal of the chemical groups such as methyl group or acetyl group. Each consists of 2 carbon, 3 hydrogen and 1 oxygen atom. The chemical group influence how tightly the DNA is wrapped around the histone, which affect whether a gene can be turned on or off. Error in the epigenetic process such as modification of the wrong gene or failure to add the chemical group to a particular gene or histone can lead to abnormal gene activity or inactivity. And altered gene activity, including the caused by the epigenetic error, is a common cause of genetic disorder. Conditions such as cancer, metabolic disorder, and degenerative disorder have been found to be related to epigenetic error. Scientists continue to explore the relationship between the genome and the chemical compounds that modify it. In particularly, they are studying the effect of epigenetic modification and error have on the gene function and protein production and human health. Now let's see the diagram of epigenetic modifications. Chromatin remodeling refers to the dynamic rearrangement of chromatin structure to regulate access to the DNA by transcription factor and other protein. It involves in the activity of chromatin remodeling complexes which use ATP to slide, eject or restrict nucleosomes. So, chromatin exists in 2 states, euchromatin that is the active state and heterochromatin that is the inactive state. Remodeling allows gene to transitions between these states affecting gene expression. Major chromatin remodeling complex include SWI, SNF, ISWI and CDH families. DNA methylation. DNA methylation is the addition of the methyl group to the cytosine residue in the CpG island region, which is the high frequency of CG dinucleotides. So mediated by the DNA methyltransferase, it repress the gene expression by preventing transcription factor binding or recruiting repressive protein, and it plays a role in the genomic imprinting, X chromosome inactivation and slicing repetitive element. In abnormal DNA, methylation is linked to disease like cancer. Histone modification. Histone protein around which the DNA is wrapped are post-translational modification to regulate chromatin structure and gene activity. Common modification is acetylation, methylation and phosphorylation. Ubiquitination and summorylation are other modifications. Now, these all modifications are common modification of histone modifications. So, histone code hypothesis: the combination of histone modification from a code that influence gene expression and chromatin state. Interplay between these processes. These mechanisms are interconnected. Chromatin remodeling can expose or hide region for DNA methylation or histone modification. Histone modification can recruit protein that either promote or inhibit chromatin remodeling or DNA methylation. Together, they maintain cellular function, development, and adaptability to environmental changes. And in context of epigenetic modification, we will see in epigenetic modification, chromosome remodeling, DNA methylation and histone modification are the key mechanisms that regulate the gene expression without altering the DNA sequence. These modifications are heritable and reversible, making them important for the development, cellular differentiation and environmental responses. So in chromatin remodeling in epigenetics, it involves the restructuring of chromatin to either expose or hide DNA region influencing gene expression. The chromatin remodels reposition nucleosome to regulate axis to promote, enhance and other regulatory elements. Remodeling determines whether chromatin is in an open, that is the euchromatin, or closed, that is heterochromatin state, controlling gene activity or repression. This regulation of chromatin remodeling can lead to diseases like cancer. And after that, DNA methylation in epigenetics happens. The DNA methylation is important in the epigenetics that involve the addition of methyl group to the fifth carbon of the cytosine in CpG dinucleotide. It silences gene. Methylated CpG islands in the promoter blocks transcription factor binding, slicing and gene expression. That is the role of the splicing gene in it. And developmental regulation also, it is essential for the process for like genomic imprinting and X chromosome inactivation. In genome stability also, it prevents activation of transposable elements and maintains genomic integrity. Abnormal methylation pattern, example hypermethylation of tumor suppressor gene, are common in cancer. If we see the histone modification in epigenetics, histone modification include post-translation changes to histone tails such as acetylation, methylation, phosphorylation and ubiquitination. So acetylation, it promotes gene expression. Role in epigenetics of acetylation is to promote gene expression by neutralizing the positive charge of the histone losing chromatin. Methylation can either activate or repress gene depending on the specific histone residue modified. Histone modification recruits specific reader protein that interrupt the histone code and guiding transcriptional machinery or repressive complexes, coordinated with DNA methylation to reinforce gene slicing or activation. Interconnecting in epigenetic regulation, DNA methylation and histone modification. The DNA methylation recruit methyl CpG binding domain protein which can interact with histone deacetylase to establish a repressive chromatin state. And in chromatin remodeling and histone modifications guide chromatin remodeling to specific region, influencing nucleosome positioning. Epigenetic memory. Together, these modifications form a memory system, ensuring that specific gene expression pattern are maintained throughout the cell. And in health and diseases, we know that epigenetic modification are implicated in numerous conditions, including cancer, neurological disorder, developmental diseases, etc. This was all about the epigenetic modifications. Thanks for watching. Do like, share, and subscribe.